We've all been hearing about viruses a lot given the current circumstances, but have you ever wondered what exactly is a virus and why do they even cause us problems? In this video we will be exploring exactly this. First things first, contrary to popular belief, unlike bacteria, viruses are actually non-living organisms. They are much smaller and much simpler than single-celled microorganisms such as bacteria. So much so that if bacteria had eyes, they wouldn't be able to see viruses with their naked eye. Viruses lack most of the criteria required to be classified as a living organism, the most important being that they lack the ability to replicate out of their own capacity. Living organisms must also be able to respond to stimuli and perform metabolism. Metabolism is producing energy. Viruses cannot do any of these. Now that we have a basic understanding of what a virus is, let's look at how a virus is structured. A virus is simply genetic material in the form of single-stranded RNA or double helix DNA enclosed within a capsid. This capsid is then enclosed within an envelope which has protein projections stuck to its outer surface. These projections act as receptor molecules and are very important for the virus. This is pretty much all a virus consists of, which compared to a bacterium looks very simple. It's very important that we know that viruses lack the molecular machinery needed to replicate. Unlike living organisms, viruses do not contain ribosomes, or any of the enzymes needed to replicate the genetic material. This is exactly what makes viruses so harmful. Viruses replicate by entering a host cell and hijacking the molecular machinery of the host that enables it for genetic replication. Viruses inject their genetic material into the host for it to be replicated, thereby forcing the host cell to make copies of the virus instead of what it would be normally doing. Now that we understand why a virus needs to infect a host cell, let's talk about how a virus infects a host cell. First of all, a virus must enter a host organism. If it's a human, they could enter from many points of entry such as the nose, mouth, a cut, or even your eyes. Once the virus is inside, it must reach its target cell. A virus can't just go and hijack any cell from any organism or animal. This is because of the protein projections on the outer surface of the virus that act as receptors. These receptors have a very specific shape, and only certain cells have the correct complementary receptors to the viruses to allow the virus to enter the cell. Think of it like a lock and key. The virus is the key and can only open the lock of a cell which fits perfectly to its key. This is why certain viruses are only able to infect certain organisms and certain parts of that organism. For example, coronavirus only attacks the lungs of humans because that's the only place the receptors on the virus are able to work on the receptors of the host cell. Once the virus locks onto the host cell's receptors, it either injects only its genetic material into the host cell, or the whole viral molecule enters the host cell via a process called endocytosis. The virus then disassembles inside the cell, leaving only the genetic material intact. The viral DNA is then transcribed and translated in order to give rise to a new viral molecule. The genetic material acts as a set of instructions that instructs how to create more viral molecules. Therefore, instead of the whole cell reading its own genetic material, it reads the virus's genetic material and produces new viral molecules. When enough of the new viruses are produced inside the host cell, the virus exits the cell via exocytosis. This often causes the host cell to burst or just die. These new viral molecules then go on to infect and kill more cells inside the host, eventually hijacking and destroying enough cells inside the host organism that they begin to feel unwell. These are the basic fundamentals of how a virus infects a host. But if we go into more detail still, there are actually two different mechanisms by which a virus actually replicates once inside the cell. These two are called the lytic cycle and the lysogenic cycle. The lytic cycle is a very quick way for a virus to affect a host. This is when the virus causes the host to exclusively replicate the viral DNA. So the host doesn't do any of its normal functions, but instead it only produces new viruses. This causes an exponential growth of the virus inside the host, with the number of virus molecules doubling approximately every 20 minutes. The viral molecules then eventually burst out of the host cell and cause it damage or death. The second method, the lysogenic cycle, is when upon entering the host cell, the viral DNA is simply incorporated into the DNA of the host cell. The host cell is able to carry on normally and replicate itself normally, while the viral DNA stays mostly silent for now. However, as the host cell replicates, each subsequent cell arising from the infected host cell will also carry the viral DNA within its own genome. The viral DNA is only replicated after a certain stimuli occurs. This stimuli could be a certain temperature or after a certain time period. This will cause all cells in the organism carrying the viral DNA to begin producing new viral molecules all at once, which is equally as bad as the lytic cycle. The viral molecules carry on infecting cells inside the host organism until they are ultimately transferred from one host to another host. The way a virus is transferred between hosts varies. Some may be transferred via a sneeze, some via a cough, or some even via food and drink. This is how a virus works.